Hi everyone, welcome back. Today we are going to discuss about tolerance and autoimmunity. This is Mia Acuña, your reporter for today. So in the topic 1, we'll be talking about the tolerance, and in the topic 2, we will be discussing the autoimmunity. So tolerance refers to the specific immunological non-reactivity to an antigen resulting from a previous exposure to the same antigen. While the most important form of tolerance is non-reactivity to self-antigens, it is possible to induce tolerance to non-self-antigen. When an antigen induces tolerance, it is termed tolerogen. Now, tolerance by definition is the prevention of an immune response against a particular antigen. For instance, the immune system is generally tolerant to self-antigens, so it does not usually attack the body's own cells, tissues, and organs. However, when tolerance is lost, disorders like autoimmune disease or food allergy may occur. Tolerance is maintained in a number of ways. So one is tolerance to self-antigens. So we do not usually mount a strong immune response against our own self-antigens, so a phenomenon called self-tolerance. When the immune system recognizes a self-antigen and mounts a strong response against it, autoimmune disease develops. Nonetheless, the immune system has to recognize self-MHC to mount a response against a foreign antigen. Thus, the immune system is constantly challenged to discriminate self versus non-self and mediate the right response. Another is induction of tolerance to non-self. Tolerance can also be induced to non-self or the foreign antigens by modifying the antigen. By injecting the antigen through specific routes such as oral, administering the antigen when the immune system is developing, and etc. Certain bacteria and viruses have devised clever ways to induce tolerance so that the host does not kill these microbes. For example, patients with lepromatous type of leprosy do not mount an immune response against Mycobacterium leprae. Now, let's talk about the central versus peripheral tolerance. Induction of tolerance requires education of both B and T cells, which occurs in both central or the bone marrow and thymus, and the peripheral, which can be found in the spleen and lymph nodes, lymphoid organs, and tissues. So here, lymphocytes become either immune competent or tolerant towards encountered antigens. And the mechanisms of tolerance induction and maintenance differ between B and T cells and between central and peripheral lymphoid organs. Now, by definition, the central tolerance is a type of immune response or immune tolerance that occurs in the thymus and bone marrow. It is the main mechanism that helps the immune system to discriminate self from non-self, while the peripheral tolerance occurs in the peripheral tissues and lymph nodes. It operates as a secondary mechanism to ensure the deletion and conversion of self-reactive T and B lymphocytes to anergenic state. Next is the peripheral T cell selection. So the central and peripheral tolerance occur in tandem in the case that central tolerance is not completely effective, partly because not all autoantigens are expressed in the thymus. Now, there are mechanisms of peripheral T cell tolerance, which includes the clonal deletion, ignorance, energy, and immune regulation. So tolerance mechanisms can also result in inappropriate tolerance to non-self antigens, and as an example, when the LMC or the LCM virus was inoculated into mouse embryos, adult mice mounted no immune response to LCM and were chronically infected. It was thought that these antigens induced early during gestation were handled as self-antigens, thereby inducing negative central T selection. Now, for the clonal deletion is the best studied mechanism eliminating activated T clones is activation-induced cell death. So T cells activated by antigen-presenting cells such expressed in IL-2 and IL-2R for autocrine facilitation of proliferation. Activated T cells also increase their expression of death receptors and their glands, and ligation and FAST leads to T cell apoptosis via the caspase pathway, thereby ending in the immune system. So clonal deletion, in other words, eliminates the harmful B cells from the lymphocytes and then induced to self-antigens, and then the process goes on to binding of self-antigens, ending in the immune response. Next is ignorance. So here in ignorance, although peripherally T cells from healthy individuals can reach with self-antigens in vitro, this does not commonly occur in vivo. It is thought that T cells ignore certain self-antigens because they are located in immune-privileged sites or because they have low immunogenicity, or immunogenicity meaning low levels of expression or low binding affinity, right? In our previous lessons. So the immune system is not normally exposed to ocular antigens, but trauma to a single eye releases autoantigens that activate autoreactive immune cells, leading to severe granulomatous inflammatory of both eyes. Another is the anergy. Anergy is the major mechanism in activating peripheral autoreactive T cell clones. Anergic T cell clones cannot respond to cognate antigenic stimuli because they do not produce IL-2 or IL-2R. Multiple proposed mechanisms explain this block in T cell activation and the disruption of the interaction between the T cell co-receptor, or the CD28, as you can see there, and the APC co-stimulatory molecule, the CD80 over 86. Now, the interaction of CTLA4 with CD80 over 86 negatively regulates the T cell activation, and the T cells displaying the CD28 tends to be activated by the APC, while T cells displaying the CTLA4 tend to become anergenic, as you can see there in the picture. So under the physiologic conditions, the T cells express CD28 on initial encounter with APC, and shortly after such stimulation, they start displaying CTLA4, which has a higher binding affinity than CD28 for CD80 over 86 than does CD28. Now, CTLA4 knockout mice develop profound lymphoproliferative disease. Another is the immune regulation. So, the immune regulation is achieved by the action of TREGs. The picture there is not very clear because I would like to commend Google for being the source of this picture, and I just use a background remover so I could get the picture that I look forward to. So here in the immune regulation, anyway, TREGs are important in the maintenance of peripheral tolerance. So when they are depleted from mice, for example, the autoimmunity results. A human patient with genetic TREG dysfunction develops lymphadenopathy and inflammatory infiltrates consisting of autoreactive T cells in multiple organs. So naturally occurring thymus-derived TREG display anergenic properties in vitro, but it can also suppress CD4 plus CD25 T cells in vivo via direct cell-to-cell contact or secretion of cytokines. So during active inflammation or infection, in other words, TREG do not prevent protective immune function. So the induction of CD4 plus CD25 in TREG can also be activated in peripheral lymphoid organs and suppress immune responses via anti-inflammatory cytokines rather than direct contact. So that is all for our tolerance topic 1. Now let's move on to our topic 2, which is the autoimmunity. So here, before we move on with our autoimmunity discussion, let us first take a look at the innate immunity and adaptive immunity. So in our innate immunity, we have there our macrophages, the natural killer cells, the dendritic cells, neutrophil, eosinophil, and basophil. And on our adaptive immunity, the B cell, antibodies, T cell, CD4 plus T cell, and CD8 plus T cell. Now, the T cells and the natural T killer cells assist both the innate and adaptive immunity of our body to prevent us or to protect us against infections and inflammations. Now, our part 2 or topic 2 starts the autoimmunity discussion. So by definition, autoimmunity can be defined as breakdown of mechanisms responsible for self-tolerance and induction of an immune response against components of the self. Now, the effector mechanisms in autoimmune diseases, both antibodies and effector T cells can be involved in the damage of autoimmune disease. Now, genes, immune system, and the environment where the patient lives are just some of the effector mechanisms in autoimmune diseases. So the diagnosis of autoimmune diseases is based on symptoms and detections of antibodies and or very early T cells reactive against antigens of tissues and cells involved. So antibodies against cell or tissue associated antigens are detected by immunofluorescence. Antibodies against soluble antigens are normally detected via ELISA process or radioimmunoassay, and in some cases, a biological or biochemical assay may be used, such as Graves' diseases and pernicious anemia. And now for the treatment of our autoimmunity diseases or autoimmune diseases, the anti-inflammatories such as corticosteroids and immunosuppressive drug therapies such as cyclophosphamide, azathioprine, and cyclosporine is the present method of treating autoimmune diseases. Extensive research is being carried out to develop innovative treatments, which includes anti-TNF, alpha therapy against arthritis, feeding antigen orally to trigger tolerance, anti-idiotype antibodies, antigen peptides, anti-IL-2 receptor antibodies, anti-CD4 antibodies, and anti-TZR antibodies, and etc. The goals of treatment of autoimmune disorders are to reduce symptoms and control the autoimmune response while maintaining the body's ability to fight infection. So treatments vary widely and depend on the specific disease and symptoms. That is all for today. Should you have any questions, suggestions, or clarifications, feel free to ask me personally, or you can ask me via Facebook, that is Acuña. So again, thank you for your participation, and until our next discussions.